Thank you, everybody, for joining us today for today's joint webinar series with GlycoNet and ACS Carb Division. My name is Pumtiwitt McCarthy. I'm an associate professor at Morgan State University. And it's a pleasure today to introduce two speakers today. Our first speaker will be Professor Mingdi Yan. Professor Yan is a chemistry professor at the University of Massachusetts Lowell. Her current research includes perfluoroaryl azide-based conjugation chemistry, glyco-nanomaterials, and gold antimicrobials. She obtained a BS from the University of Science and Technology of China and a PhD from the University of Oregon. The title of her talk today is Carbohydrate Conjugated Fluorescence, Turn on Azide Probes for Imaging Cells and Bacteria.
Well, thank you, Professor McCarthy, for the introduction. And I would like to start by thanking the organizer for inviting me to this GlycoNet and ACS Carb Division, the joint webinar. So today I'm going to talk about fluorescent turn-on imaging probe based on azide and its carbohydrate conjugated to image mammalian cells and mycobacteria. So the azide probe is based on this perfluoroaryl azides. My group has been working with this type of azides for 2 decades now, long time ago, as a nitrine precursor for conjugation material functionalization, and also as electrophile to react with dipolarophiles in cycloaddition reaction, and also nucleophiles. So there are many unique reactivity and properties for this type of azides, primarily because of fluorine on the arrow ring. And I will show a couple of examples later. And also fluorine facilitates characterization, for example, by fluorine NMR and by XPS. They are quite straightforward to prepare. They are quite stable. So when we first started to study this group of azides, I often get questions about how safe it is to work with azides. Do you get explosion? Ever since the click chemistry comes along, and I don't get this question anymore. In terms of the electrophilicity of the azides, we have summarized our work in this recent Account of Chemical Research article. You can see this type of fluorinated aryl azides can undergo a variety of reaction with nucleophile and the dipolarophiles. And most of the time, we actually don't use metal catalysts, and some reactions occur at room temperature without any catalysts. For example, the first reaction here with enamine to give the amidine at room temperature, very high yield, and without the fluorine, you get the triazoline. So it's a different reaction pathway. And the reaction with aldehyde, either by organocatalyst or in the presence of the base, we get the amide as a product. Again, very mild condition. And the last reaction here is the Staudinger reaction. This reaction, for one, is very fast. It's one of the fastest Staudinger reaction. And for two, the product stops at the phosphorimine. It doesn't undergo hydrolysis reaction like other Staudinger reaction. So it stops here, and because of the fast kinetics, and the reaction allow us to label cells, as you can see here, cell surface labeling with sugar. Because of the fast kinetics, the background is quite clean, you can see. So all this property is because of the fluorine introduced on the arrow ring that give us a very nice handle. So today I'm actually going to use the other property of the aryl azides. That's the photochemistry of the aryl azides through nitrine. So Matplots has spent almost his lifetime study the photochemistry of aryl azide, and it's very nicely summarized in one of his book chapter and here. So if you start with aryl azides under photoactivation, it excites form the excited states, and the splitting the nitrogen will give you the singlet nitrene. And the singlet nitrene is actually the branching point for all the subsequent reactions. Without the fluorine, it can undergo many different reactions: ring expansion, protonation, intersystem crossing, and of course the productive routes, which is CH insertion and CC double bond addition reaction. So the aroazides actually was used since the late 60s, early 70s as the photoaffinity labels in biochemistry to study the binding site structure of receptors. With the fluorine, this so-called protective pathway, the yield is much more improved. Sometimes you see a reported yield of 96%. And it's because the fluorine increase the lifetime of singlet nitrene and suppress the other pathways by increasing the activation energy. So we have used the photochemistry of the aroazides for conjugation surface functionalization, for example, with polymers and the sugar. In this case, it's the so-called glyco-nanomaterials. And we also work on carbon materials, nanotubes, buckyballs, and recently graphene. The other unique thing about this type of molecule is that you can introduce another group, making this a heterobifunctional. And then this group can work on a variety of different materials and surfaces. So it's quite versatile. So today I'm going to talk about another unique thing about this kind of aroazides. That's the photoactivation, fluorescent turn on. And more than 10 years ago, the Merler group has used this fluorinated aroazides as a single molecule imaging for live cell imaging. So the way it works is that the aroazides, it's non-fluorescent until you turn it on by photoactivation. So photolysis undergoes the decomposition of the azide into singlet nitrene, which abstract a proton or hydrogen from neighboring molecule to give you the aniline, and the product becomes fluorescent. So today, the molecule I'm going to talk about is this carbazole version of the fluorinated aroazide. Under light activation, it undergoes intramolecular CH insertion, right, into the neighboring CH bond. And the product, again, is fluorescent, where that the reactant is very minimally fluorescent. The molecules are fairly straightforward to make. So we start with the methyl ester of a pentafluoral methyl ester, commercially available, relatively inexpensive, and convert that into the azide in high yield. And then the subsequent another substitution reaction with the carbazole give us the product. Because there is a handle here, methyl ester, we can turn that into activated ester, and a simple amide formation with a mean functionalized sugar to give us the carbazole. In this case, it's mannose-derivatized, the carbazole derivative. So the intramolecular CH insertion, like I said, quite efficient. This is the NMR conversion, greater than 90%, and 68% is isolated yield. We confirmed that it is indeed the intramolecular CH insertion, not the hydrogen abstraction in the case of W.E. Merler's case. So we made this amine derivative by reduction. And then you can see from NMR, they are very different. They also have different photophysical property as well. We monitor the conversion. So the absorption, when we see the absorption of the product increase and the reactant decrease, this is the delta absorption of the product and the reactants. And you can see after photoactivation, the emission has increased, in this case, more than 40 times. And then we measure some of the photophysical, photochemical parameters. here again, this number one is a W. Merler's molecule. The lambda absorption, lambda max, is 407. Ours is on the shorter wavelength because we don't have this conjugation. But the substitution on the carbazole does change the lambda max here. The other thing that I wanted to bring to your attention is that the half-time, the fluorescent turn-on half-time, is much shorter, and then in the case of Muller's molecule. So you can see it several times faster. And the last parameter is this so-called photoconversion quantum yield. So it measures the azide transformation, or the conversion from azide to amine per photon, per each photon absorbed. And ours is at least 10 times higher than Muller's molecule here. So it's very high efficiency in terms of the CH insertion reaction by intramolecular pathway. With that, so we did cell imaging with this Manos derivatized probe. And of course, for this audience, I don't have to say too much about Manos. It's transported to a mammalian cell with facilitated diffusion mediated by this group family of the membrane transporter. So our question is, can this large carbazole derivative be tolerated by cells, be transported? So we did some preliminary characterization on the confocal microscope. So here's a slide. We have 4 spots here. We have the sugar derivatized and the non-sugar derivatized. This is the starting materials, and those are the photoactivated product. You can see after activation, so this is actually increased 30 times after, before, after. So we can say that after UV activation, the fluorescence increased about 30 times. So that's a good preliminary data allowing us to carry on the cell imaging. So here is on the HUVEC cells. So here's the setup. So we have the sample on the stage, and then we come along with LED fiber, UV fiber to activate, to turn on. So you can see the fluorescence increase with the irradiation time. And you can see the cells are all light up in this case. Here we compare the one with Manos and without Manos. And then you see before UV and after UV, the fluorescence is much stronger after irradiation. And interestingly, after 24 hours, the Manos derivative somehow diffused, whereas the non-Manos derivative still stay. We then, stain the HUVEC cells with Lysotrack, and the merged image indicates that the compound, the Manos derivatized compound, actually was co-localized in the lysocell. So that was the cell imaging. And the next, I'm going to show you another example where we derivatized this fluorescent turn-on azide probe with Trehalose. Yeah, so before I get into the results, I want to say a little bit about mycobacteria and the TB diagnosis. The TB diagnosis, there are multiple ways to diagnose TB. Of course, the gold standard is the cell culture, where you culture the mycobacteria tuberculosis. But it's this bacteria, it's very slow growing, takes days to grow, and so it's very time consuming, but it is the most accurate. So you have TB. And of course, the latest Gene Expert, it is a PCR based. It can detect mycobacteria tuberculosis and the rifampin resistant mycobacteria tuberculosis. And it's a sputum sample, and it's very fast, right? You can get results within a couple of hours. It has high sensitivity, specificity. However, for resource limited regions, for developing country, it is still expensive, and it also requires skilled personnel to carry out the assay. And in the developing country, still a popular technique is also called smear microscopy. It's an acid-fast staining technique. It's, again, an older technique. It's based on the fact that mycobacteria, it's resistant to acid stain. So in this example here, you can see the mycobacterial tuberculosis is stained red among the blue background. Again, so it is a cumbersome and a long process. It requires a lot of sample processing and staining and so forth. And the most problematic aspect of smear microscopy is that its sensitivity is quite low. Somewhere between 20 to 80%, especially in the HIV co-infected patients, the sensitivity is quite low and also require quite a bit of bacteria in order to be detected. So new technique that can facilitate the detection and early diagnosis of TB, it's still is still needed. So one of the technique takes advantage of trehalose. So I'll give you a couple of examples later. So trehalose, again, for this community, I don't have to say too much. It is in mycobacteria, very specific for mycobacteria, especially in the cell envelope. It is the glycolipid component, right, which make the membrane very resistant to antibiotics. And the mode of action involves the antigen 85, and which is very unique to this organism. And it is also essential to the pathogenicity of mycobacteria tuberculosis, which causes TB. The nice thing about trehalose is absent in mammalian biology. And lastly, it is taken up by both the mycobacteria tuberculosis and the mycobacteria smegmatis, and that is the non-pathogenic strain used by us and by many other people for model study. All right, so here's one example of using trehalose-based imaging probe to detect and image mycobacteria tuberculosis by Clifton Berry and Ben Davis. So they made a lot of trehalose derivatives, and they have shown one of the key message from this paper is that this AG85 can tolerate virtually any chemical modification on the trehalose. So basically AG85 catalyzed the trans-esterification reaction to turn the trehalose dimycolate into trehalose monomycolate in this example in here. And then they conjugated FITZI onto a trehalose. Trehalose, you can see they have beautiful images showing that this probe is taken up primarily in the cell envelope, clearly seen here. And most importantly, they also are able to image or label only the mycobacteria that is infected with the macrophage. So in other words, this trehalose probe is very specific for mycobacteria in the presence of macrophage or mammalian cell, very specific, which is highly desirable. Here's another recent example by Bertozzi's group. There they use this so-called solvatochromic probe. So this probe is non-fluorescent in aqueous environment, but in lipophilic environment, for example, in micromembrane in the mycobacteria, fluorescence is turned on in this hydrophobic environment. So it's very useful because then you don't have much background. Yeah. This probe detects viable bacteria, and here they have the image of smegmatis, and actually they made a FITZI derivative of the trehalose. In their case, they didn't see very high contrast, unlike the example I just gave you by Clifton Berry and Ben Davis. All right. So So our question when we had this probe is that can we use the trehalose-based fluorescent turn on, the azide probe, to image mycobacteria, especially this, again, this very large molecule, can it be tolerated by mycobacteria? So we went ahead and made the trehalose derivative. Again, very straightforward chemistry, several steps to install a amine group on the trehalose. So we didn't make this complicated, just to proof of concept, and then conjugating onto the carbazole derivative through the NHS ester to give us the trehalose derivatized, the carbazole azide probe. Again, the fluorescent turn on, just like we tested before, it's pretty straightforward and clean. And it's fast as well. So this is absorbance of the reactants decrease very fast. And then the fluorescence of the product is, in this case, is 19 times higher than the reactants. So there is this fluorescent turn on. And property just as we have seen before. Again, the parameters here are very similar to the previous probe we have before. Very fast photo conversion, rate of photo conversion, and then the also the conversion quantum efficiency is quite high. So then we check some of the conditions for cell imaging. So literally here, what we did is that we took Mycobacterium magmatis and we incubate with our carbazole trehalose probe. And then we use a handheld, this is the TLC lamp, literally at 365 nanometer. And we turn this on and measure the fluorescence of the sample. So you can see, even with incubation time, this is like, yes, those are like 20 minutes or 10 minutes. It's already over 50% of the fluorescent intensity is turned on. Yeah, irradiation time, even at 30 seconds, you see quite efficient in terms of the fluorescence of the product. And this is the concentration study. Not quite the final optimized, but and that allow us to go ahead and see whether this can be used to image mycobacteria. So we did first did some the mechanism study and see which pathway the probe is taken up by mycobacteria. So on the left here, we mix our probe with trehalose, the free trehalose, a different ratio. You can see the more trehalose, free trehalose we have, the lower the fluorescence intensity. So that tells us that it is hijacking the trehalose uptake pathway. And we did another assay, resotrophin assay. You start with the butyrate. It's a very nice assay developed by Don Roning. It's a fantastic assay. So butyrate is not fluorescence. After transfer catalyzed by Ag85, the product is fluorescent. So we use this assay and we increase the concentration of our carbazole derivative, the turn-on probe, and you can see the higher the concentration, the higher the fluorescence. That means that Ag85 is involved in the uptake in our case as well. We use another assay to further prove it's Ag85 dependent. In this assay, it's abscilling, so it is a Ag85 inhibitor. So the more abscilling or higher concentration abscilling, the lower the fluorescent intensity, meaning that really Ag85 it is involved in the uptake of our carbazole probe. We then checked the uptake on the heat-killed bacteria, and it doesn't taken up much at all. So that tells us the uptake is active uptake. It needs the bacteria to be alive. Finally, We checked on other gram-negative and gram-positive bacteria, the negative of Pseudomonas aeruginosa and E. coli, and the gram-positive Staphylococcus epidermidis. In all cases, you can see the fluorescence is minimal. So that tells us that the uptake is very specific for mycobacteria. So finally, I want to show you the imaging, the confocal imaging. These are still preliminary, not the most beautiful images, but it did show that the fluorescent turn on, it is indeed can be used to image mycobacteria. So the first panel here are before light activation, and then we turn it on by just irradiating with the handheld TLC lamp for minutes at 365 nanometer. So you can see they are exactly the same location, right? So you can see highly fluorescent bacteria after photoactivation. This image on the right here, it's the image when we treat the mycobacteria with the product. In other words, we activate the Trehalose probe first. So it already does the CH insertion, so the probe is already activated. Then we use that to treat the mycobacteria. You see the fluorescence is much weaker. Again, we need to collect more images, but it does tell us that our fluorescence turn on is much more efficient and has high contrast than this is basically conventional fluorescence probe. Again, this is very latest data. We need to repeat, but it is very encouraging. So here we are checking the detection limit. So we start with 10 to the 8th and then dilute log-wise, and 10 to the 7 and all the way to this is like around 15 bacteria. I don't know if you can see, still you can see some faint fluorescence here, and this last row is bacteria only. So what we did is we mix our compound with the bacteria at different concentration. And then we use the handheld TLC lamp and irradiate, and then we clean it thoroughly so we don't have residual the compound left. And then this is the image taken by my student's cell phone. This is the fluorescent intensity taken by the plate reader. And then you can see that, so correlate pretty well with the imaging data on the left. Yeah, so with that, I want to summarize to show that our fluorinated aroazide and the carbazole-derivatized azide can undergo intracellular, intramolecular CH insertion reaction to give a fluorescent product. And they are quite straightforwardly synthesized, and the yield is pretty good, and the photo conversion is fast and efficient. And I've also shown that the carbohydrate derivatized carbazole imaging probe can be tolerated and taken up by both mammalian cells and bacteria. And they have given high signal intensity and contrast on conventional fluorophores. And we think the mode of action of the Trehalose probe is that it hijacks Trehalose uptake pathway, and it's AG85 dependent, and it's active uptake, and it's specific for mycobacteria. And going forward, we are planning on to do the experiment with TB, the real TB, and infected and TB as well. So with that, I would like to thank the people who did the work. And this project is literally initiated by my former PhD student, Shen Xie, a very talented student. And it's now picking up by Sajani Liyanagi, and she did all the bacterial work. And the imagings are taken by Dr. Earl Ada and Dr. Ying Fu. And I didn't have time to talk about some other work. For example, our work on the Trehalose nanoparticle to detect mycobacteria in collaboration with Bob Langer and Anna Jacknanek at MIT, and the rest are some of my current collaborators, and my group before the pandemic. The funding is primarily from NIGMS, and with that, I thank you for listening, and I look forward to our Q&A session.